Good afternoon, and welcome to today's webinar sponsored by NAVBO's Education Committee. I am your host, Dr. Benicio de Jesus Perez, and I will be moderating today's session. We're pleased to welcome our speaker, Dr. Paolo Madeddu from Bristol University in the United Kingdom. We'll also like to welcome Dr. Yev from Dr. Madeddu's lab. She will monitor today's questions. Now, before we begin, I would like to remind you that all NAVBO activities are now easily found in the calendar of events on our website. So please be sure to register for some of the upcoming events. Throughout the presentation, please type your questions in the question box in the control panel. These questions will be answered at the end of the presentation. After those questions, provided there is time, the attendees will be able to ask any additional questions in real time. If you have a question, raise your hand by clicking the hand icon on the left side of your control panel. You will be recognized, unmute your mic, and ask your question. This webinar is being recorded and archived on the NAVBO website for future use. Now, to talk a little bit about our speaker, the overall goal of Dr. Madeddu's present and future research consists of developing more effective strategies to treat limb and myocardial ischemia and to improve the current methods employed for correction of cardiac defects in infants. His own contribution has been the identification of novel angiogenic factors, human tissue kallikrein, and nerve growth factor. In addition, he's looking at successful genetic programs that support health rather than trying to contrast pathogenic mechanisms. Currently, Dr. Madeddu and his lab are conducting research on microvascular damage in COVID-19 with a focus on spike protein-induced signaling in human parasites. So with that, I would like to welcome Dr. Madeddu.
Thank you very much, Vinicius, for this kind of presentation. And I will go to my first slide, which is repeating the title of my webinar. So, reverse cardiovascular aging by the use of longevity proteins. Also, illustrate this nice bridge, which was built in Bristol a century ago. And I use this bridge just to exemplify a method to translate basic research to real medicine for the benefit of patients, which is actually my mission. So this particular webinar will focus on heart aging and will highlight new approaches to support cardiovascular fitness and health as one ages. One major problem for doctors and in general for our society is the increasing prevalence of older people. So what is called the aging equation. So one side, we have a decline into the state of increasing frailty by an increasing number of people. And you can see here depicted as a slow decline in health from literally difficulty in walking to the state of being confined into bed. So this kind of aging equation is also a klaxon call of distress for policymakers and financial modelers, who are desperate to balance the equation of supporting the demographic change with a voracious healthcare need. So our society is spending a lot of resources and not only financial, but also human resources to try to balance this wrong equation. And if you wanted to have more information about this problem and the financial burden and the economic, but also not only economic, societal imbalance, you can just refer to this website link. So most of the complications that are associated with aging and regards the cardiovascular system are illustrated by these two slides. So one is heart attack that are more frequent in older people, and then the progressive chronic decline of the heart function that lead to heart failure. These are probably the more clear clinical evidence of the state of disease. But obviously, there are other aspects of a cardiac aging that can be described as a progressive decline of function that lead to inability to the patient to have a normal life. So how today these conditions are treated? We have actually powerful drugs that are able to decrease cholesterol and decrease blood pressure to control risk factor, and also to reduce the symptom in this aged patient. We have also interventional procedure, such as, for instance, angioplasty, which can be performed by the doctor, by the cardiologist or the surgeon, to reopen an occluded vessel, and so to reduce the damage of vessel occlusion. But all these kind of measures are only palliative, because they cannot replace the damaged cell when they are irreversible injured. And obviously, they are also quite expensive, so can be afforded to a larger population, but not all the people are suitable for the treatment or available, have available treatment in the place where they live. So particular poor place in our society are somehow discriminated by the fact that this sophisticated procedure cannot be delivered. So what is the benefit of this expensive treatment is that people live longer, but they live with a permanent damage. So we have people that are have their acute disease transformed into a chronic disease. And I think that I needed to recall some reflection for you now, basically because when we're talking about the heart, we need to also consider some limitation intrinsic to this particular organ. As you know, there was in the last year a big argument about the possibility of the heart to have intrinsic mechanisms of regeneration, such as, for instance, stem cell that allow the heart to regenerate. You are familiar with the initial hypothesis provided by the group of Piranversa, and also the polemic against his first papers. But certainly, even the heart can have a number of stem cells, the potential, the regenerative potential of this cell is very limited. So the heart is very different from other organs in human body, in the sense that one damaged the capability of regeneration is not sufficient to replace the damaged cells. So what we can do actually to encourage the heart to heal, apart from using available drug or performing angioplasty and other revascularization procedure. So I'm quite interested by the possibility to interfere with biological and chronological aging. But before talking about this approach, I wanted to illustrate what is a biological clock and a chronological clock. So chronological age is the amount of time that one person passes from birth to a given date, and is your age in terms of years, months, days. This is the primary way people define their age. But the point is that not all people age the same way. So the aging clock is different from person to person. So we need now to define what is instead biological age. So biological age is different from chronological age because it takes into consideration a number of factors other than just the day you are born. People can appear younger or older than their age, and we are interested why this happens and how can be exploited to prevent age-related diseases. So here are some examples of actual age versus heart age. We have an example, for instance, of a 40 years old male. He's a smoker, he has a high blood pressure, he's diabetic, and have a normal body mass index. But actually, all these conditions make him having a real heart age of 75. We have now a 50-year-old female. She's not a smoker, but she has also high blood pressure. She's also diabetic and obese with a very high body mass index. Now her real age is 85. I can say that to be optimistic, there is also people that is 45 and they demonstrate less than that, or 55 and demonstrate maybe 45. And I think that if you try to recall your experience, certainly you can see people where you say, oh, yeah, look very young for your age. Okay, now we will try to zoom into the aging heart and zoom more and more deeply, starting with the heart as an organ. So it's normal that we say with aging, the heart tend to change. You don't need to have ischemic heart disease. The heart remodel, and the reason is that with time, some cardiomyocytes die, and the only way to replace them is the deposition of collagen and the increase in the volume of the ventricular chamber. We can try, obviously, to reverse this phenomenon, or at least to delay this phenomenon by exercise and other environmental changes. But if we zoom now to the level of the cell inside the heart, you can see that the main alterations that accompany aging are fibrosis, so deposition of collagen between the cardiomyocyte, infiltration of inflammatory cell. Not all of them are necessarily negative. Some really simply remove the garbage, or eventually also perform some reparative reaction responses in the heart. And also, this alteration in cardiomyocyte performance lead to diastolic dysfunction, which means the heart is less able to relax. At an intracellular level, if we look at the organelles inside the cardiomyocyte, we can see that the mitochondria don't work as they supposed to do, and there is an increased oxidative stress and energetic imbalance from their dysfunction. There is also in the aging heart an increased apoptosis and the loss of regenerative capacity. I would say, in particular, reduced angiogenesis. In addition to that, with time, some alteration at the chromosome level occur, particularly shortening of the telomere and the somatic mutation. And this accumulated together with the tendency of some cells of the heart, particularly the stromal component, to secrete the inflammatory factor. Epigenetic change also occur, and change in the scenario of non-coding macroRNAs. So one of the most clearly described alteration in the aging heart, even in the absence of myocardial ischemia, is what is called heart failure with preserved ejection fraction, which is illustrated in this cartoon. You can see on the left a normal heart, and on the right a heart with heart failure and preserved ejection fraction. You can immediately appreciate that there is an increase in the size of the ventricle, but mostly because of accumulation of collagen, which increases the stiffness of the ventricular wall. In addition to that, there is an impaired angiogenesis, vasoconstriction, and loss of endothelial function, which lead to coronary vasoconstriction. When we zoom at the level of molecular mechanism, you can see here that the inflammation is associated with and capillary rarefaction, together with endothelial dysfunction, are associated with increased oxidative stress, reduction of nitric oxide production, and then this induce additional activation of fibroblasts and accumulation of collagen, which basically make more distant the blood flow from the cardiomyocyte and induce a relative ischemia. I'm zooming more into the organelles inside the cardiomyocyte, just to describe that during aging, you can see there is a remodeling of the mitochondria, which are the subcellular structure that produce energy, but this energy is associated with increased production of oxidative stress. This, let's say, oxidative stress garbage increase with age and lead also to damage of the DNA contained in the mitochondria, which lead obviously to alteration, permanent alteration in mitochondrial function. Another organelle that is much affected by aging is the proteasome system, which basically is a mechanism by which the cell tend to eliminate protein, unfolded protein. Protein are progressively tend to accumulate in unfolded fashion into the cell, and there is a mechanism that eliminates this unfolded protein. But unfortunately, this with time this response is with time overwhelmed, and this leads to protein accumulation and damage in the cell, the cardiomyocyte. Another organ that is attracting much attention is the nucleolus. The nucleolus is how to say, is a little less recognized organelle in aging, but it's a last very recent research point out on the fact that this organelle is affected by aging in different way, particularly in the ability to control the ribosomal biogenesis as a production of new ribosome. So it's also important in makeup of RNA and the protein, and so is its damage lead to altered protein synthesis and ribosomal function. So coming to possible new approaches that to combat the cardiac aging, there are several options now, and still at the experimental stage. Some have been translated already in initial clinical trial. One in particular on the left here, the use of omecamtiv, which is a novel selective cardiac myosin activator. This is quite attractive because should increase the contractility of cardiomyocyte, but unfortunately the first clinical trial didn't reveal much improvement if compared with traditional treatment. Also, as a matter of particular attention are antioxidant and vitamins, because they say are supposed to contrast some molecular mechanisms that are altered in the aging heart. But so far, there is very little evidence that antioxidant or vitamins can really delay the aging of the heart. And there's also the question how safe these treatments are, particularly the high dosage of intake. The other approach that is trialed recently is consists in the use of senolytic agent. We spend a few words on this because they attract much attention, and the reason is that they're supposed to eliminate the senescent cell and leave just cells that function well. But unfortunately, in the case of the heart, this is not very rational, because as you kill cardiomyocyte, you remain with very few, maybe a little bit younger than the ones that are eliminated, but I don't think this is the right strategy for the terminally differentiated organ like the heart. Probably for other organs, but not for the heart. So the rest of my presentation will focus on another alternative approach, which is capturing the top secret from healthy centenarians, which are healthy and lucky individuals that survive very long and are extremely part of the Gaussian curve of survival. Some of these centenarians are particularly interesting because they not only live longer, but live longer and healthy. So they have sometimes diseases such as diabetes, but they don't experience any complication of diabetes and other chronic age-related diseases. So I will spend 2 words about mutation, and I will talk and try to distinguish between negative mutation and positive mutation. In this slide, I show an example of negative mutation. So we have a pressure from the environment, and this butterfly basically are pressured by the fact that if they are yellow, they will, their destiny is to be eaten by birds. So mutations that lead to brown butterfly exposed to less risky. And this because there will be a pressure for increases the number of brown butterfly. A similar example is given by people living in a particular area and exposed to risk factors, such as, for instance, malaria or other environmental problem. Some mutations can prevent or protect them from malaria, but unfortunately, they also expose to other risk factors. Example, for instance, is people living in Africa that were deprived of salt, so they developed mutation they allow to save salt. But when they emigrated to other area where salt was available, they were exposed to high blood pressure risk. So they is called the salt-sensitive hypertension in Black Americans. So this is a negative pressure. But now examine the possibility of a positive pressure mutation. So these are basically the case of all the people. They are exposed to a good environment, and some mutation allows them to live longer. But this positive mutation does interfere with increase in prevalence of the mutation. So the frequency of the people with this mutation remains the same inside the population. But they represent a kind of a positive element for this particular population environment, because their offspring will inherit this particular mutation and continue to represent a minority of people that live in healthy condition. They also influence the environment because basically they are able to pay attention to particular aspect of the environment which reinforce the fact to be old but healthy. So it is supposed that if you can discover some of the secret or gene secret from these long-living people, we can eventually try to fight all the diseases that affect all the people in the general population. There are several groups in the world that they are studying all the people, all the population in order to capture this secret. For instance, I mentioned just one of these studies that were performed in the US. In Albert Einstein College of Medicine, they are collecting the record of people that live longer and compare with people with typical normal lifespan. And also they collect information from the offspring to understand what is the difference between these different populations. But as I said before, we are not only interested to understand this gene, but also to exploit the secret to deliver some benefit to the general population. So if we understand what are the proteins responsible for longevity, in particular acid longevity, we'll posit that we be able eventually to transfer this genetic information through gene therapy or protein therapy to all the rest of the population. And now I will give you a typical example of this strategy, the ones that we are performing in our laboratory. So we are interested in this particular gene, a mutation of the human BPIFB4 gene, which encodes a secreted protein initially found in salivary gland and olfactory epithelia to confer microbial resistance. The downregulation of protein of the olfactory system, such as BPIFB4, is a nasal mucus, contributed to age-related decrease in olfactory function, which is considered a very strong predictor of degenerative disorder and death in the elderly. In previous publications starting in 2015, we showed the polymorphic variant of the PBFB4 associated with exceptional longevity under recessive genetic model in 3 independent populations across the world. A more rare mutation instead leads to reduced activity and risk of cardiovascular disease, in particular hypertension. So there is a kind of gene titration that lead to completely opposite phenotype, one healthy and the other diseased. In this presentation, I will focus on the healthy mutation. Here is just the frequency of the gene mutation. You can see the geographic disparity, with East Asian showing the highest prevalence, as then the North Europe, while the Southern Europe and Africa have the lower prevalence. We then checked what is, we performed 3 parallel study, clinical study, to understand what is if there is an association between the mutation and cardiovascular disease. The first study was done in a population of elderly subjects that were referring to the diabetic center in Padua, in North Italy, and we evaluated the association between the circulating level of the protein and the presence of myocardial ischemia. You can see here the having high level of the protein lead to a lower risk of having cardiovascular disease, and in particular myocardial ischemia. So the risk is reduced by almost 20% relative probability. Then we look to a second population, people with recent acute myocardial infarction, and again we tested the level of the protein in the circulation. Again, the presence of a high level of the protein reduces the risk of having S3 vessel disease by almost 14% in terms of relative risk. And finally, we tested a third population. We compared a patient which undergo heart transplantation versus donor of the heart. The two populations were different in terms of, obviously, of age, but we confirmed the difference in terms of expression of the protein in the heart, even after correction for the age as a confounding factor, age and also sex. You can see that patients with myocardial ischemia and heart failure have a lower level of the protein, both as a level of myocyte and vascular cell identified by specific antigens. So we particularly are interested about pericyte. The pericyte are a particular focus on my lab, are cells that are surrounding the vascular endothelium, and are very important for maintain the vascular homeostasis, but also for regeneration. So we have in the lab a methodology to isolate and expand the pericyte, which we use for this particular purpose. So we obtained isolated expanded pericyte. Here you can see the typical antigens that defines this cell, and we obtained it from both donor and also from failing heart, the populations that I described in my previous slide. And in this case, we tested the pericyte from ischemic heart, where can show some form of senescence. This was the case. You can see the marker of senescence is remarkably activated in this cardiac cell, human cardiac cell. Also, there was evidence of mitochondrial oxidative stress, and in general, oxidative stress was increased. Vitamin D1 receptor was decreased, which is an indication of less regenerative potential of the cardiomyocyte. And again, we confirmed that the level of protein was decreasing in this cell, confirming the data of lower level in the circulation. The next step we tried to rescue this altered phenotype by providing a supplementation of the longevity protein, but also the wild type protein, to understand there was difference between the two isoforms. You can see that the longevity protein, so the one that was encoded by a mutated gene, was much more effective than the wild type, and obviously the vehicle, in terms of reducing the sign of senescence, also oxidative stress, and also rescuing the level of vitamin D receptor. I have to say that regarding these other two aspects, the wild type and the longevity protein perform the same way, but the longevity protein was superior for in combating senescence and oxidative stress. Then, interestingly enough, in collaboration with a group in Germany, we were looking to the ability of the protein to modify the contractility of the cardiocyte. And this was really unexpected but very interesting result. So cardiocyte derived from human iPS and treated with the LAV protein were contracting much better, so with a much higher amplitude, but also show an increased frequency. So they the protein seems to have very strong chronotropic, inotropic effect on isolated human cardiocyte. We also explored the mechanism behind this effect, and we discovered that the mutant protein is activating the phosphorylation of downstream signaling through PERK and strengthening the association with 1433, creating a kind of a scaffolding system of a phosphorylated protein, which ultimately activate the nitric oxide signaling. We also described a mechanism implicated in the nucleolus. It seems that the protein form a complex with nucleolin inside the nucleolus, and this lead to reduction in inflammatory cytokines and increased angiogenic factor. And then we perform a series of studies that I don't have time to describe, particularly angiogenesis studies using in a network formation by pericyte alone together with endothelial cell, which shows a protein increase angiogenesis in vitro. The last 10 minutes of my presentation will be dedicated to some in vivo experiment in animal models. So the first study was just to see if we were able to combat atherosclerosis in mice. And we published this in European Heart Journal 2 years ago, so you can have access to this article. What we saw that administering the longevity protein led to reduced inflammation, reduction in the number of atherosclerotic plaque, and reducing in high blood pressure in this animal. We also were quite interested about the possibility for the protein as a gene to combat diastolic dysfunction, in particular dilated cardiomyopathy, which is a typical pathology that occur in during aging in older people, but also during diabetes. And so we treated a diabetic animal with vehicle or the wild type isoform or the longevity isoform using an adeno-associated virus as a vector. And just to summarize the result, using echocardiography, we demonstrated that although the animal continued to be diabetic, they were experiencing less complication for the heart, which were consisted in improvement in the ejection fraction, less diastolic dysfunction as assessed by measuring the E/A ratio, and improve angiogenesis, neovascularization of the heart, less fibrosis, and less lipid deposition. In addition to that, it was quite a serendipitous observation, but when we eliminated the fur on the chest of this animal to do echocardiography, the untreated animal have a regrowth of the fur that was very slow, while the LAV-treated animal, where the regrowth of the fur was very, very fast, which is actually conceivable with improved perfusion of the skin of this animal. You know that this could be quite clinical interesting for diabetic people because they suffer for the loss of their hair, which is marginal in terms of survival, certainly, but aesthetically could be very important for a patient. We also performed an additional study and published both the study in 2 different journals. You can see one in European Journal Heart Failure, and this on cells. We described the fact that the administration of this longevity protein lead to substantial changes in the activity of several key enzymes in the mitochondria, leading to an improvement in the mitochondrial production of energy and less oxidative stress disposal. The second studies that we performed very recently was to give the longevity gene, again, through an adeno-associated virus in older animals. And we did actually two experiments. I just show you here one of these. So one was prevention, the other one was a rescue, so in older animal. In both cases, we saw an improvement in stroke volume, so in index of contractility, as compared with the other group of vehicle, GFP, and wild type, and consistently also an increase in cardiac output. Zooming as a level of the tissue, we saw an increase in capillary density, a decrease in cellular senescence. We also performed a positron emission tomography to assess blood flow under basal condition and following stress. And while the untreated animal treated with vehicle or wild type didn't have any improvement in myocardial perfusion following stimulation with tobutamide, you can see here the animal treated with the longevity gene had basal perfusion increased, but also they responded very well to the pharmacological stimulation. I remind you that the absence of increasing perfusion means that the ability of the heart to increase the coronary flow is basically abolished, which is a case of a treated animal. We are restoring completely not only the basal level of perfusion, but also restoring the ability of the heart to increase perfusion under stress. And lastly, we performed also an experiment in infarcted mice in which we compared the longevity protein versus the GFP, again through adeno-associated virus. You can see here that there was an improvement in stroke volume, again in cardiac output. We also obtained an increase in capillary density in the perinfarct zone, and reduction in interstitial fibrosis. So I believe that these are just a few examples. We are extending the study to progenic animal, and we have several projects ongoing on different models to build as a case for a first-in-man clinical trial, which will be performed with a recombinant protein. So I believe that it's time for me to give you some message from my perspective. So I think that it's very clear that every single one of us has an aging clock, but some very lucky ones have a clock that goes slower, and some of them instead are less lucky have a clock that go faster. So maybe we can rewind the ones that go faster if you know the mechanism. And one mechanism could be the one that is inherited by people that live longer and healthy. I have also to say that, in my opinion, this approach is quite safe as compared with the traditional drug, because traditional drug obviously have a side effect. This gene and the related protein were selected through 1,000 years of evolution and shown to be actually produce health, but also to be safe. So I believe that this protein will pass well the test of safety studies. And I want to conclude that the way these older people help the new generation is not only based on gene. Certainly very important to make a better environment and transmit their experience. So there is an enrichment that come also to the environment and next generation. And yes, I think that I agree with this cover from Times. which says that healthy aging is in our gene, but maybe also the gene transmit the benefit to the environment and help the environment to be better. And I believe that the strategy of taking some of important information from healthy elderly people is incredibly fruitful for the possibility of passing this information not only to the offspring, but also help other people to live better. And I leave you with this message and with my last slide. Thank you very much for your attention.
Thank you so much, Dr. Madeddu. That was an excellent presentation. I'm going to go ahead and start and ask the first questions before I give the floor to Dr. Yue. So you've talked to us about the heart, but I'm also thinking about the brain. I'm also thinking about the peripheral circulation. Now, this model linking genetic with senescence, do you think this is a paradigm that also applies outside the heart to other circulations that are also vulnerable to age and associated with comorbidities that ultimately also compromise longevity and quality of life of our patients? And if so, do you think by addressing them as you're proposing, you could potentially be providing a platform that will not only improve heart health, but also brain health and circulatory health?
Yeah, absolutely. I think it is a very, very important question. And my response is yes. It's yes, because we have additional experimental evidence. So, for instance, we saw benefit in model of limb ischemia, which I didn't have time to show here, but I published data. We have published data also about the ability of the protein to interfere with immunity in the positive way. So to convert, for instance, inflammatory M1 macrophage into M2 macrophages. We have also evidence that actually the protein helps the immune system to respond better to viral infection, including the coronavirus. One major application that we can see is to combat aging-related vascular decline in the brain. So one major project would be really to delay all the aspects of aging in the human body, not limited to the heart. But obviously, because of the mission of NAVBO, I just focused today on the cardiovascular system. So it would be a general approach rather than an approach limited to a single organ.
That is great. Thank you so much. So I'm going to pass on the floor to Dr. Yue, who will tackle some of the questions that we have in the chat. Dr. Yue, the floor is yours.
Okay, here we comes a question from Kishore, and he asked, I'm wondering how long AAV9 continue to express in the gene in these mice? And Prof. Paolo?
Yeah, thank you very much for this question. Basically, we performed an experiment, as I said, in two different model of aging. We started with mice that have an age that corresponded to, let's say, 50 for human, and also another experiment in which the corresponding age was, let's say, 65. And what is most interesting is in both cases we got positive result. In case of very old animal, we were able to recoup the function of the heart by something like the corresponding of 10 years, which is really extremely good if reproduced in humans. Regarding the need of giving different dosage and how long a single administration lasts, remember that we use an adeno-associated virus, which allow for prolonged expression in the heart and different organ. We found expression in the liver, but we also found, because this particular adeno-associated virus was targeted to the liver, we found also protein expression several weeks after the injection in the heart. Now, with the protein, the situation will be different because we'll need to administer the protein several times, so basically every day. I can tell you that in mice, the administration of the protein was able to reduce blood pressure in hypertensive rat, to decrease blood pressure for about 3 days. So one single administration should be sufficient to reduce high blood pressure in animal for let's say 3 or 4 days. But eventually we need to re-administer the protein because half-life and particular pharmacodynamics of the kinetics of the protein.
Okay, thank you. And here's another question from Catherine. And she asked, wondering if you believe that the benefits you're seeing in multiple models, including atherosclerosis and with love, is primarily due to improved endothelial performance, communication?
I'm sorry, but I didn't capture completely your question. So you mean difference between the wild type and love?
Well, I think she wants to know between the multiple models with love is primarily due to the improved endothelial performance or the communication. So the yeah.
Maybe even can help because I really.
So let me unpack that a little bit. I think the point being here, so you talk about endothelial cells, you talk about pericytes, and you started your presentation giving us a broad view of the cellular milieu in the heart. So I believe what Dr. Ho is pointing out is, do you think that the benefits that you see in vivo is related to the fact that you're potentially targeting endothelial dysfunction, normalizing cell behavior, and ultimately the capacity of endothelial cells to interact with other neighboring cells like cardiomyocytes and pericytes? I believe, I don't know if Dr. Ho can comment in the chat if I got the gist, but that this is my interpretation.
So. Okay. Yes. Thank you. Sorry for not capturing immediately the question. I think this question also we received from the reviewers. So I think it's very important. So I think that the effect of the benefit is come obviously from different side. So it's very difficult in vivo to discriminate what is coming first and what guide the benefit. When you are giving anything to a complex organism, the response are heterogeneous, but is also sometimes synergistic. So to discriminate between the different cell benefiting more from the approach, we did several studies in vitro, and we found that, as I mentioned before, an improvement in contractility of the cardiomyocyte, in isolated cardiomyocyte, which indicates that the protein, and we have data showing that there's an improvement in calcium handling by the cardiomyocyte. Also, we saw that, particularly in the diabetic model, there is the expression of fetal protein that are associated with a better contractility performance by the cardiomyocyte. So certainly there is a direct effect on cardiomyocyte. But when given into the organism, the whole organism, certainly the effect on angiogenesis, improvement in endothelial function, together with better performance of the pericyte, resulted in better perfusion, as I show in one of my slides. So this indirectly can improve the cardiomyocyte function and reduce apoptosis. And obviously, in this case, also the fibrotic remodeling. So, as I said, we believe that is an overall effect on several cells, and which is can be dissected in vitro, but also quite clear in vivo.
Okay, Prof. Here's we come another question. He he asked, there are many studies with the results similar to yours on the effects of the epicatins. Are you aware of this?
About aware of the
effects of the epicatins. So we have some similar results.
So you mean about the tachycardic effect?
I think the Dr. Rubio is asking about the epicatessins. Apparently, he points that there are other studies that have looked at the epicatessins, and are reporting similar findings. So I think he wants you to comment on how your experimental design models and assessment compares to other, what other groups have reported.
Yeah, obviously we are not, we are quite excited with our result, but we believe that there are other ongoing research on focusing on senescence and other anti-aging treatments that can eventually improve the health state of the heart and other organs. I believe that, for instance, in terms of reciprocal support, I'm not negating, for instance, the possibility of combining this approach, actually. So I don't see any competition. Instead, I can see possible synergic combination of a treatment, for instance, a senolytic activity, senolytic drug together with treatment and longevity protein in combination could be eventually work even better. But before engaging in comparison, and obviously comparison are very important because you need to compare with a competitive strategy. But in this stage, I believe that it's very important that each laboratory continue research, and exchange information in order to implement all the possibility, and then eventually compare and go to possible combination. I not negate the possibility of combination. And the other thing that are very important that we are able to exchange and publish obviously having intellectual property, because it's very important to involve industrial actor in this research. But on the other side, also sharing as much as possible the information between lab in a way that we can eventually accelerate the process. As a doctor, I mean, I'm a cardiologist in background. I believe that now we reach almost the top in terms of potential of all the fashion drug, and it's time to just focus on a new approach to treat aging-related diseases. So again, I believe that I'm very, very interested about comparing, but in this moment, I would be quite prudent in say one is better than the other one.
I will certainly agree with that, and I think the scientific field benefits from different perspectives and obviously transparency and rigor. And ultimately, we have to think of how these discoveries translate to therapeutics. And the more information we have about mechanisms and complementary models, the greater the chances that this will potentially be translatable into something that could improve the quality of life and survival. So I agree with that, and I think ultimately it's a good lesson that goes beyond what we're discussing here today, but applies to science in general. There's no, there's no monopoly on one pathway, one molecule. So anyway, I'm going to ask if there's any other questions before we wrapped up. I don't see anything in the chat, but if anybody has any question, yeah, so I think otherwise, I'm going to go ahead and wrap up. If you have a question, please email info@navbo.org and we'll make sure to share your questions with Dr. Madeddu. So I do hope you enjoyed today's seminar. Again, I want to thank Dr. Madeddu for an excellent presentation. Uh, for all of you, please join us on Thursday, April 7, 2022 at 1 p.m. Eastern Time for our next webinar presented by Dr. Karen Christman from the University of California in San Diego. The title of her talk is Injectable Extracellular Matrix Biomaterials for Translational Regenerative Engineering. Please look for more information on the NAVBO website. And thank you so much for joining us today. We appreciate your visit. I hope everybody stays safe. Again, thank you, Dr. Madeddu. Thank you, Dr. Yue.
Thank you.
I'll see you all next time. Take care. Bye-bye.
Bye.
Bye.